What if the traits you inherit from your parents were determined by a simple set of rules? Let's journey back to the 19th century, where one man unlocked the secrets of heredity, revolutionizing science forever. Welcome back to Fact or Theory. Today, we'll dive into the groundbreaking work of Gregor Mendel and his laws of inheritance, which changed the way we understand genetics. Mendel. A brief overview. Gregor Mendel was not your typical scientist of his time. Born in 1822 in what is now the Czech Republic, Mendel was a man of humble beginnings. He grew up in a farming family, which laid the foundation for his lifelong fascination with nature and plants. Despite financial struggles, Mendel pursued an education, eventually joining the Augustinian monastery in Brno. This move not only provided him with the resources to study but also shaped his scientific discipline. The monastery was not just a religious institution but a hub of intellectual activity. Mendel's scientific journey began with a question that many overlooked at the time. How are traits passed from one generation to the next? To answer this, he turned to the humble pea plant. Why peas, you might ask? They were easy to grow, reproduced quickly, and exhibited clear, contrasting traits like tall or short stems and yellow or green seeds. Mendel's meticulous nature and the simplicity of these plants made them the perfect candidates for his groundbreaking experiments. Between 1856 and 1863, Mendel conducted thousands of experiments in the monastery garden. He cross-pollinated pea plants, carefully documenting the traits of their offspring. What he discovered would later be called the laws of heredity. Let's look at them one by one. Laws of heredity. The law of segregation is the cornerstone of Mendel's discoveries. Mendel observed that each individual inherits 2 copies of a gene for a given trait, one from each parent. These gene copies, which we now call alleles, segregate during the formation of reproductive cells, gametes. As a result, each gamete contains only one allele for each gene. For example, Mendel worked with pea plants that had either tall or short stems. When he crossed a pure tall plant with a pure short plant, the offspring in the first generation, F1, were all tall. But in the next generation, F2, the short trait reappeared in about 1/4 of the plants. This consistent 3-to-1 ratio demonstrated the predictable nature of heredity and confirmed the principle of segregation. This law is crucial because it explains how traits don't blend but instead retain their integrity across generations. Today, we understand that this segregation happens during meiosis, a type of cell division that produces gametes. Next, the Law of Independent Assortment expands upon Mendel's findings. This principle states that the inheritance of one trait is independent of the inheritance of another, provided the genes for those traits are located on different chromosomes. In simpler terms, the color of a pea, green or yellow, has no bearing on its shape, round or wrinkled. To test this, Mendel performed dihybrid crosses, where he tracked 2 traits simultaneously. For example, he crossed plants with round yellow seeds with those having wrinkled green seeds. The resulting offspring exhibited all possible combinations of traits, such as round green or wrinkled yellow seeds, in a consistent 9-to-3, 3-to-1 ratio. This law explains the incredible diversity seen in living organisms and is a fundamental concept in understanding genetic variation. It's also why siblings can inherit completely different combinations of traits despite having the same parents. The third key insight Mendel uncovered relates to dominance and recessiveness. Mendel found that some traits dominate over others, meaning that if an organism inherits both a dominant and a recessive allele, only the dominant trait will be expressed in the phenotype, the observable characteristic. For instance, in Mendel's experiments, tall stems were dominant over short stems. Even if a plant inherited one allele for tall stems and one for short stems, the plant would appear tall. The recessive trait, in this case, short stems, only appeared when the plant inherited 2 recessive alleles. This discovery was revolutionary because it challenged the prevailing idea that offspring were a blend of their parents' traits. Mendel showed that recessive traits don't vanish but remain hidden in the genetic code, ready to resurface in future generations. These laws were groundbreaking, but they also had limitations. For example, Mendel's work didn't account for traits influenced by multiple genes, polygenic traits, or environmental factors. However, his principles remain foundational, forming the basis of modern genetics. Today, the law of segregation, law of independent assortment, and the principles of dominance and recessiveness are applied in diverse fields. They guide genetic research, help in understanding hereditary diseases, and enable advancements in agriculture, such as creating disease-resistant crops. Conclusion: Mendel's brilliance lay in his ability to distill complex patterns into simple, universal laws. His work transformed biology and inspired countless scientific breakthroughs. So next time you consider the traits you've inherited, from eye color to height, remember Mendel's pea plants and his meticulous experiments. His insights continue to influence science in ways he could never have imagined. Thank you for exploring Mendel's laws with us. If you enjoyed this journey into the foundations of genetics, hit the like button and subscribe for more deep dives into science and history. See you in the next video.